In this video, we will go over how to passage cells. You will need phosphate buffered saline, also known as PBS, 0.25% trypsin. This can be found in the -80°. In order to open the flasks, open the top one first, followed by the second. This will avoid moving over them and possibly contaminating them. Aspirate out all of the media in both flasks. Do this by tipping them to the side and aspirating on the side with no cells on it. Rinse with 5 milliliters of PBS in both flasks. This is dependent on the size of the flask. Our flasks are 75 centimeters squared, so we use 5 milliliters to completely cover the bottom. Slowly rinse the bottom of the flask by oscillating back and forth with the PBS inside. Aspirate out the PBS. Add 5 mL of PBS again and repeat the rinsing process for a total of 2 times. Add 3 mL of 0.25% trypsin to each flask. Before adding, mix the trypsin with a pipette because it was previously frozen and some of the particles could be stuck at the bottom. In order to mix the trypsin, pipette some of it up into the pipette and then pipette it back down into the solution. The trypsin causes the cells to detach from the bottom of the flask. The amount of trypsin is dependent on the size of the cell flask. With the 75 centimeters squared cell flask we use 3 mL of trypsin. Oscillate the cell flask from side to side to make sure that the trypsin completely covers the bottom of the flask. Now place the cells into the 37°C incubator. The incubator will speed up the trypsin process. Leave the cells in the incubator for roughly 3 minutes and then examine under a microscope. The cells will look like little round balls when they are all detached. The rest of the video will be shown using one flask. Now, place 3 mL of media onto the trypsin. This will prevent the trypsin from harming the cells any further. Mix by slowly oscillating the cell flask back and forth. When removing cells from the flask, pipette some of the liquid and then inject it down onto the bottom of the flask. Find a systematic approach to this. Repeat several times. Next, pipe up the entire solution and transfer into a 15 milliliter centrifuge tube. Now take the cell solution and centrifuge it. Be sure to balance the centrifuge with a centrifuge tube of equal volume. Centrifuge at 180 Gs for 7 minutes. After centrifuging, you should be able to see a small white cell pellet at the bottom of the centrifuge tube. These are all of the cells from our cell flask. Slowly aspirate out the media and trypsin mix. Be very careful of the cell pellet at the bottom. Tipping the centrifuge tube as much as possible makes this easier. Now add 2 milliliters of media to the cells in the centrifuge tube. After adding the 2 milliliters of media, be sure to pipette up and down gently to mix the cell pellet. After mixing the cell pellet, remove 2 milliliters from the centrifuge tube and add a milliliter to each new cell flask. Now add 12 mils of media into each cell flask. Be sure to oscillate the flasks back and forth so that the media completely covers the bottom. Place the cell flask back into the incubator and label with your name, the date, your PI's name, and the cell line, as well as the passage number.